Some definitions in enzymes. The molecule that the enzyme is changing is called the substrate. So we looked at this thing right here is the enzyme, and it's labeled right here. This is what we called the reactant before. And so when we wrote these equations before, we said something like A is broken down into B plus C. And so in this case, A is sucrose. It's a sugar. And so here's two monosaccharides bound together here by a bond, a covalent bond. And so this is a disaccharide. And look, it's getting broken into two monosaccharides. So if this is A, then this would be B, glucose would be B, and fructose would be C. So that's the equation that's happening. So here is the reactant, and then B and C, these things, are called the products. So you should already know the terms reactants and products. Here's what you need to know now. The reactant is also called the substrate. So it's called a substrate when an enzyme works with it. So if you just have this equation, A becomes B plus C, and there's no enzyme involved, then you call this the reactant. If there's an enzyme that you need to speed up this chemical reaction so A is broken down more easily, then instead of calling it a reactant, you can also call it a substrate. So a substrate is the chemical that the enzyme works on. So here you can see the substrate and the enzyme together. So that's called a substrate enzyme complex. And that's here. And so the enzyme will just bend this enough so that it's easier to break, and that's how they'll break. So stress is placed on the bond by binding of the substrate and enzyme, and the bond is broken. And then you have these products, B and C, in this case, glucose and fructose. And then the interesting thing is, look at this enzyme. Now it can be used all over again to do the reaction again and again and again and again, and again. Okay, active site, another definition. Most enzymes are proteins, and they're folded into tertiary and sometimes quaternary structures. So if you remember with proteins, you've got a primary structure, which means one amino acid bonded to another amino acid, bonded to another amino acid, bonded to another amino acid, and so on. And then they fold into these alpha helices or beta pleated sheets, sometimes like this. And so these would be all kinds of beads, all kinds of amino acids linked together, and they would be kind of folded like that. The tertiary structure is when they fold over on top of themselves like that, and quaternary would be if you have more than one of these polypeptide chains together. So because enzymes are proteins, they can get folded into really special, unique shapes. If you have just one amino acid different, then the whole shape of the protein could be different. So the enzyme's shape is really, really important to see whether it will fit or not with the substrate. And so here's an enzyme. This is the part right here called the active site where the substrate will fit. So they're folded, the protein or the enzyme's folded conformation, that means its shape, creates an area known as the active site. It's a special region of the enzyme molecule which binds to the substrate. And so here's the enzyme. This thing right here, that space is where the substrate or the reactant will fit. And so therefore this spot on the enzyme is called the active site. So that's the active site. And look at how the substrate fits. Whoops. Look at how the substrate fits right in. Perfect. So the substrate joins, I'm sorry, I keep flipping ahead. The substrate, back. Okay, here I am. So the substrate fits right in with the enzyme. And there you see it's the right shape to fit right in. Okay. So the enzyme substrate coming together will make the product. So here you have the substrate, and here's the enzyme. Here's the enzyme substrate complex when they're together. The enzyme stresses this substrate just enough so that the bonds break. Sometimes it goes the other way, but right now we're breaking bonds. And now you have these two different things that are going to be released. These are the products. So these are two products. And here's the enzyme all ready to go back and do it all again. Or maybe I should draw it down like this. So it's going to keep going and working again and again and again. So enzymes keep on working. Specificity. So that sounds like the word specific, and that's what it means. It means that each enzyme is specific for just one type of substrate. So the nature and arrangement of amino acids in the active site make it specific for only one type of substrate. So for example, if this substrate is, let's say, the sugar lactose. So hopefully you remember that ose means that it's a sugar. So lactose is a disaccharide. Let's say this enzyme is lactase. Oops. And enzymes end in ase. And so this enzyme fits just perfectly with this substrate, and it will break it down into its two monosaccharides. And this enzyme will work with lactose again and again, and another lactose, and another lactose, and another lactose, but it will not work with any protein. It won't break down lipids. It won't even break down a different sugar. So it's very, very specific just for lactose. Sucrase, for example, that's ACE, means an enzyme, will break down only sucrose, which again is a sugar. Lactase will only break down lactose. Each enzyme is the right shape and charge for just one substrate. So by shape and charge, I mean, if you look at this, it fits just like a puzzle right here, and so on for the whole shape. The other thing is, if this is slightly positive here, this will be slightly negative here. If this is slightly negative, this will be slightly positive. So it's not just the shape, but it's also the charges. They'll also be attracted to each other. The body, the human body, or really any living thing, could be bacterium even, requires thousands of different chemical reactions to live. And therefore, there are thousands of different enzymes in the body. There are enzymes that break things down, like we just were talking about sucrase that will break down sucrose. There are also enzymes that will build molecules up and make them bigger. One enzyme type is present for each type of chemical reaction. They are reusable, super important. You make the enzyme once and then it's used again and again and again for a long time. So you only need small amounts of enzymes because they're used over and over and over and over again. And here's a really great video, I think, of it. So this red thing is the enzyme, and here's the substrate or reactant. Catalysis means it's breaking it down, it's catalyzing, and here it's breaking it down, and look, you have the products, two things. And the thing is, you can, whenever you get a new substrate, the enzyme will work and do it again and again. Here's a new substrate, up, it's breaking it down to more products. Here's a new substrate, it's going to happen again and again and again. One other really interesting thing about this is something called the induced fit, which I'm going to talk about soon. But if you look at this, it's not exactly the right shape yet, and so that's how we think enzymes probably work. The substrate will actually change the shape of the enzyme enough so that it works. So the initial fit is a little bit different until the substrate actually goes in, but they're still super, super specific. Enzymes are not permanently changed in the process, so they're used again and again and again. They are reusable.